Welcome to the latest Novogene webinar, Introduction to Transcriptomic and Epigenomic Solutions. I'm John Cafferty, Marketing Manager here at Novogene Europe. This is our first webinar after the summer break and the first in our new autumn programme. We'll send you full details of the upcoming webinars and hope you can join them for them. One I would like to mention is the Novogene European User Forum, which takes place on the 7th and 8th of October. We have a fantastic lineup of speakers for this, and places are limited, so make sure you register early. It will, of course, be a virtual event, so you can enjoy it with a cup of tea from the comfort of your desk or kitchen table if you're working from home. On to today's webinar and a bit of housekeeping. The webinar will last around an hour with a 10-minute question and answer session at the end. Please submit your questions via the Q&A button you'll see at the bottom of your screen. You can do this at any time during the webinar. We endeavour to answer your questions live, but if we can't fit them all in, we'll get back to you by email after the webinar. I'd like to introduce you to our host for today, Reza Javan. Reza completed his doctoral degree in clinical medicine at the University of Oxford. He was based at the Big Data Institute, where he developed various bioinformatics software. His PhD research involved using large-scale genome mining approaches, which led to the discovery of hundreds of new viruses, as well as 14 potential new antibiotics. He joined Novogene in January and is currently a regional account manager for Oxfordshire and South Wales. Reza? If I can unmute you, Reza, hold on.
Hi guys, can you hear me?
We can now, thank you.
Fantastic. Thank you very much for the introduction, John, and a very warm welcome to everybody joining us in our webinar today. As John mentioned, my name is Reza Javan, and I am a regional account manager for Oxfordshire in England and the south of Wales. Before we dive into this webinar, I would like to give you a brief introduction to our company. So, as you probably already know, Novogene is a leading provider of genomic services and solutions. And ever since we started, our mission has been, and still is, to improve life science research and human health. To tell you a little bit about our journey, it all started just over 9 years ago by a very ambitious bioinformatician in Beijing, where our headquarters is still based. And ever since then, we have been growing at an exponentially fast rate. Despite being a relatively new company, we now have multiple labs and subsidiaries all around the world. To name a few, we have a couple of labs in China. We have a lab in the other side of the world in California. We have a lab in Singapore. And in 2014, we started our UK lab. And very soon after that, we had to relocate to somewhere much larger in Cambridge Science Park, which is where it is based now and where we process samples from our European clients. At the moment, we are the largest sequencing center in the world. In fact, the cumulative output of our instrument is over 280,000 whole human genome sequences per year. And to process this tremendous amount of data, we have our very own supercomputing and data center. We perform our sequencing using the most advanced sequencing platforms currently available. In terms of short-read sequencing, for example, we have several Novogene 6000, which is the most powerful Illumina machine, along with HiSeq X, also by Illumina. We also provide our services using PacBio platforms. In fact, we are the largest user of PacBio in the world. And we can also provide sequencing services using Oxford Nanopore technologies. In terms of the actual sequencing that we do, The short answer would be pretty much anything you would expect from NGS being able to offer. Just to name a few, in terms of DNA sequencing, it doesn't really matter if your genome of interest is a human genome or animal genome or plant genome or microbial genome. We have a wide variety of different services to be able to help your research. However, the focus of this webinar today is on our transcriptomic and epigenomic services, which in fact I will not discuss, as you will hear shortly from my colleague who will describe it in a lot more details. But before I introduce you the main speaker for today, I would just like to very briefly tell you 6 reasons why I think Novogene is a great collaborator for your NGS needs. Number 1 is quality. If you have ever worked with Novogene or know anybody who has, they will probably tell you that we take quality extremely seriously. There is a quality control in every step of working with us, and we will provide you with many detailed reports as we process your samples. Also, for almost all of our sequencing, we guarantee the quality and the amount of data that you will receive. For instance, for Illumina sequencing, we guarantee that over 80% of your reads will be Q30 or above, which is even more than Illumina's official guarantee. Next is capacity. We are, of course, the largest sequencing provider in the world, capable of sequencing thousands of genomes a day. But we are also fiercely flexible when it comes to the type of project that we can accept. It can range from a single tiny microbial genome resequencing to sequencing whole genome of 10,000 people as part of a national population genomics project. In terms of technology, we have the latest available platform, and we are always in the lookout for any new technology that may become available. Also, you may have heard about Falcon. In case you haven't, there is a very cool video on our website that I encourage you to check out, which shows this platform. Essentially, Falcon is a very intelligent robot that works 24 hours a day and automates the whole process from extraction to library prep to sequencing and so on, which allows us to process your samples at a much faster rate and minimize any potential human error. And with the economy of scale on our side, or extensive experience and modern platforms, we can provide you with a very fast turnaround time. In terms of analysis, of course, this is encoded in our DNA. Our founder was a bioinformatician. And in fact, nearly half of the employees of this company come from a bioinformatics background, just like myself. So we very much understand the frustrations that bioinformaticians face, and we always have the end result of your project in mind. We also have a lot of different bioinformatic packages that we can offer you. And finally, it is our commitment to innovations. We understand the field that we are in. Things change very quickly in genomics. And for us to stay number 1, we need to continuously innovate. And this is evident from over 150 software copyrights that we hold and nearly 30 NGS-related patents. Our services are trusted by over 4,000 universities and institutes and biotech companies who come from 5 different continents and over 60 different countries. And the data that we produce for our clients are frequently featured in very prestigious journals such as Nature or Science and so on. And there's even a section on our website where you can check out the latest publications where we have generated the data or have done the analysis. But I think one of the main reasons we are known to have a long list of long-term clients comes down to our fantastic sales team. We have 25 representatives in Europe who are there to help you no matter where you are in your project. And all you have to do is to simply write an email to this address, europe@novogene.com, or go to our website and make an inquiry, and you will hear from one of our representatives depending on where you are based. So if you are based in University of Oxford, I will be in touch with you. If you are in Copenhagen, for example, we have a representative who speaks Danish. If you're in France, we have multiple reps who speak French and so on. So that's, I think, already enough from me. I would now like to introduce you to our speaker for today, who is our Senior Product Manager, Dr. Wei Li. Wei received her master and PhD from Clemson University in the United States, where she worked on the cotton transcriptome. After her academic career, she started in our bioinformatics department as a senior bioinformatics engineer. And very soon after that, she was promoted as our current product manager. Last year, around this time, Wei was giving lots of seminars and talks in conferences, I believe around 5 different countries a week, which was a great success, but I do not think it's gonna happen anytime soon again, given the current pandemic situation. But Wei is a fantastic educator and a great source of knowledge, and I'm sure you will appreciate her talk today. Without further ado, Wei, I am going to stop hugging the mic and pass it on to you.
Thank you for the introduction, Reza. Now I'm going to share my screen. Can you all see my screen?
Yep, we can. Thank you.
Okay. So thank you all again for joining us for the Novogene webinar. And for today, I will give you an introduction for transcriptomic and epigenomic solutions provided by Novogene. So in my presentation, there will be 4 parts. First, I will go over our service workflow very briefly, and then I will move into the details for our transcriptome services and epigenome services. And in the last part, I included some case studies to show you how we can support you in your research. In this slide, you will see our general workflow for RNA-seq services. So here we start with the total RNA and move down to the library preparation, sequencing, and bioinformatic analysis. As Reza mentioned, for each step, we will run very strict quality control for your sample, your library, and your data. So even though you may target different type of RNAs, like long non-coding RNAs or messenger RNAs, so it doesn't matter too much to us. You can only send us your total RNA, and we will take care from here. So to prepare your total RNA, you can always contact us for extraction services. Or if you prefer to extract your RNA in your lab, you can always use those recommended methods based on your sample information or sample status. Before you send out your samples, we highly recommend you to run some quality control before you ship it out. So here I listed some methods that you can use for your samples. You may use the gel to check if there is any potential contamination, such as protein contamination or DNA contamination, or if there is any degradation in your samples. Or you can use nanodrop because this is very common for most of our labs. And you can check the RNA purity by using those ratios or quantify your RNAs a little bit. And you can also use Agilent instrument to check another important parameters for your RNAs. That will be the ring number. This is the RNA integrity number we will use to assess how severe or the degradation level of your RNA. So this number will range between 1 to 10, and the higher, the better. So after you check your DNA quality and send that to us, so we will run the quality control again for samples as well. And after we have that result, we'll move on to the library construction step. So in the middle, I listed the major library tabs we will use for different types of RNAs. For messenger RNA, eukaryotic messenger RNAs, they are quite common to share the poly A tails. So poly A tail enrichment will be our default method to enrich your messenger RNAs for your messenger RNA-seq projects. For other non-coding RNAs in the eukaryotes or for the prokaryotic RNA-seq, we usually will use the ribosome removal method to enrich those RNAs. If you recall the pie chart I showed you in the previous slide, ribosome RNA will take almost 90% of the total RNA in our samples. And by removing them, it will be very efficient for us to enrich those RNAs. And on the right, I listed the general steps we will use for the library preparation. And here I use the poly enrichment method as an example. So usually we will enrich our target RNA, such as here we use the poly A tail method, and then we fragment them into small pieces. Then we perform the reverse transcription to generate the first strand cDNA, and then followed by the second strand cDNA synthesis. Then we will move down to the end repair, A-tailing, and adapter ligation. With that, your library template will be ready for the PCR enrichment. So after the PCR enrichment and the quality control, we will load those RNAs onto the sequencer. So if you have your own preferred method to construct the RNA libraries, you can always send us your pre-made libraries to us. But before you send out, please make sure you confirm with us your library is compatible with our sequencer platform. So as Reza mentioned, we relocate our Cambridge lab to the Cambridge Science Park in 2018. And for all of our European customers, you will send your samples or libraries to our UK lab, and we will perform majority of our projects in the UK lab if this is what you need from our sequencer. So for our UK lab, we usually can process the projects for whole genome sequencing, whole exome sequencing, RNA-seq, and pre-made libraries. And with that, for our advantages, Reza has done a very good presentation in that part, so I will not go into details. And the last step will be the bioinformatic analysis. So as you can see from this figure, even though most of our body cells have exactly the same genomic DNA within each cell, but still we are expressing different transcriptomes. So the major questions we want to address by using RNA-seq will be those two questions. We want to quantify what kind of transcripts has been expressed in our cells. So I will call that as annotation step, and to find out what are they. And the next question will be the quantification. We want to quantify, or we want to check out the abundance of each transcript. So we need to test how many of each transcript in our cells. So those will be the general steps we will include in our bioinformatic analysis for the RNA-seq services. So with that, I will move to the second part, the transcriptome services. And the first service I will introduce will be the eukaryotic messenger RNA-seq. As I mentioned, we will focus on two questions. First, gene expression quantification, and the other will be the annotation for the sequenced messenger RNAs. So this service is available in our UK lab, and we will prepare the poly A enrichment library for your projects. So in here, we will use the parent 150 to sequence your insert cDNA library, and this is the data recommendation for different species. And for the transcripts with very low abundance, we may recommend you to sequence more. So it's kind of your choice, but here we just list a recommendation for you to choose. And below will be the bottom part will be the sample requirements. So I will not go into details one by one. So if you need any detailed information for the sample preparation or the sample delivery, you can always contact our local representatives. And as I mentioned, we need to address 2 questions for the RNA-seq. And here I listed the pipeline, including the general steps we will use or we will perform for your RNA-seq projects. So here, once we got the RNA data, and I will perform the quality control on those raw data, and only the clean data or good reads will be used for the further steps. So we will map those short reads to our reference genome, and by doing that, we can answer those 2 questions I've mentioned. First, we'll perform the structural analysis, and with that, we can identify the alternative splicing events and if there is any mutation showing up in my RNA data. And the second road is the gene expression quantification. So through the mapping step, we can quantify our transcripts and perform the differential expression analysis to see if there is any genes or transcripts has been differentially expressed in my samples. And we will also include the enrichment analysis for those genes as well. To sequence the messenger RNAs, there will be 2 major methods, because for the NGS platforms, it is quite common to perform the short read sequencing. However, if you only map those short reads back to your reference genome, so it's quite possible that we don't have enough connectivity. So this may introduce some bias or some errors once we don't have the enough connectivity. So with that, we'll propose an alternative method, which is the long-read sequencing. So this will be carried by our PacBio platform. And with that, we can provide the full length of your RNAs, so that with that information, there will be no fragmentation and no PCR bias introduced during the sequencing. However, by using the PacBio platform, there will be some other disadvantages as well, because this is not as mature as our NGS platforms. So we will find out very high error rates in our long-read sequencing method. And if you really want to sequence all of your full-length RNAs, you have to make sure you got very nice RNA input materials. With that, we really need extremely high sample quality. And with only PacBio data, we cannot perform the quantification for your transcripts. So if you really want to quantify each of your transcripts, we need to use the NGS data as well. So for the long-read sequencing method, we kind of developed the service, which is the iso-seq method or iso-seq service. So here will be some detailed information for the eukaryotic iso-seq. As I mentioned, so those advantages by using the long-read sequencing will be ideal for improving annotation in the reference genome, characterizing gene isoforms, alternative splicing and fusion genes, and complementing the short or incomplete transcripts. And to sequence those data, sequence your libraries on PacBio, we'll construct the PacBio RNA library. Here is the data recommendation, and below is the sample requirements. As you can see here, so we do need very good RNA input by using the iso-seq service. So another question we may discuss a lot in the research area will be the cell heterogeneity in the bulk messenger RNA-seq method. So it's quite common in some disease studies, such as the tumor study, because in the tumor tissue, there will be multiple types of cells. And by using the bulk messenger RNA-seq, we may mask those heterogeneity of the cells. 10x genomic platforms provide a possibility to construct the single-cell RNA-seq library at very high throughput. However, it really needs a very high cell activity, and with that reason, we cannot provide the library preparation currently. However, if you prepare your 10x single-cell RNA-seq library, you can always send us your library, and we will perform the pre-made library sequencing, and also analysis is possible. So simply speaking, the single-cell RNA-seq principle is like this. You just load some input, which will be your single cells in the suspension to the platform, the 10x platform. And then they will label each cell and also each transcript with a unique sequence. So by using those information, we can quantify the gene expression profile for each of your gene from each cell. So that will be the amazing part by using this kind of library, and we can zoom in to the single cell resolution for your RNA profiles. So that will be everything I want to mention about the coding RNA or messenger RNA. And for the non-coding RNA services, as you can see from those figures, so non-coding RNAs will not be translated into proteins as messenger RNAs, but they can interact with other molecules, like they can interact with proteins or DNAs or other RNAs to get involved in the gene regulation. So in addition to the annotation and the quantification for those non-coding RNAs, we also include the target prediction. Like we want to know which RNA or which molecule is my non-coding RNA targets. So with that, I want to introduce the first non-coding RNA service, which is the eukaryotic long non-coding RNA service. In this service, we will include both long non-coding RNA and the messenger RNA information or analysis. So in this one, for long non-coding RNA, we define the non-coding RNA, which is longer than 200 nucleotides as long non-coding RNAs. And in the sequencing part, we can sequence both long non-coding RNA and messenger RNA, as I mentioned. To perform the analysis for you, we do need a reference genome and annotation available for this species. Our library tab for this service will be the strand-specific ribosome removal RNA library. And we will also use the pair and 150 method for sequencing. And here is the data recommendation. Bottom will be the sample requirements. As I mentioned, for non-coding RNA services, we will also include the target prediction in the analysis part. So if you check this pipeline, in addition to the quantification and some annotation steps we will include for the messenger RNA, we also have the long non-coding RNA target prediction as I squared out here. So that will help us to address which kind of transcripts will be my long non-coding RNA potential targets. And again, for long non-coding RNA services, we will include both link RNA and messenger RNA analysis in your package. The next non-coding RNA service I want to introduce will be the microRNA service. They have very special configuration, which will allow us to move forward for the library preparation directly. So its library will be very specific small RNA library, and the insert size will be between 18 to 40 base pairs. For the analysis, again, we do need reference genome and complete annotation. As it has very small insert size, as listed here, so we will only use single end 50 for the sequencing. And here is the data recommendation. Bottom will be the sample requirements. The last non-coding RNA service I want to introduce will be the circular RNA-seq. For this one, again, for analysis, we need a reference genome and complete annotation. And for the library preparation, this is quite special because we will include strand-specific ribosome removal and linear RNA removal steps in the library preparation. So with this extra step, we will make sure our circular RNA will be captured as much as possible. And the sequence strategy will be pair and 150. Here is the data recommendation, and below will be the sample requirements. So with those mentioned non-coding RNA services, each of them could address their potential target question. And with those services, we kind of look into the interaction between two types of RNAs. So how about I really want to find out the network between multiple types of them? So how can we get a global map? Here we develop the service, which is the whole transcriptome sequencing. For this, we will use your total RNA and split them into two parts. So first, we will use them to construct the ribosome removal and directional RNA library. From this library, we'll collect the information for long non-coding RNA, messenger RNA, and circular RNA. And the other part of your total RNA will be used to construct the small RNA library. So obviously, this will provide the information for your small RNAs. By collecting all those information, we'll perform the analysis as shown in this table. So for the standard analysis, we will include the standard analysis for each individual non-coding RNA, as I mentioned. So long non-coding RNA, circular RNA, and the small RNA. We will also provide a free joint analysis to study the networks. So the networks will include the interaction between microRNA and messenger RNA, and also the network among three types of RNAs. So with that, it will allow you to check the network or the global map or the interaction relationship between multiple types of RNAs. There will be two things I want to point out for our whole transcriptome sequencing services. First, if you check the sample requirement for each individual service, if you really want to include all of those RNA informations, you do need to prepare at least 10 microgram of your total RNA. However, if you check the sample requirements for our whole transcriptome sequencing service, we only need half of them. So you only need to prepare 5 microgram of your total RNA. That will be enough for us to construct those two libraries and collect all those informations. And the second thing I want to emphasize is, for this circular RNA library, it is not linear RNA removal library. So this really depends on your research purpose. If you really want to look into the circular RNA individually, so you may choose the previous service I mentioned for the circular RNA. So that will be the ribosome removal, directional, and linear RNA removal library. This service, the whole package, is kind of looking to the network between multiple types of RNAs. So by using this library, that will be enough for us to cover the circular RNAs. And we may lose some of the circular RNA information. However, as I mentioned, we focus on the network between multiple types of RNAs, not just the circular RNA. So with that, all the previous services will be for the eukaryotic species. And for the prokaryotic species, if you look into the individual species with reference genome, you can choose our prokaryotic RNA-seq service. For this one, we will construct the strand-specific ribosome removal library. And here is the sequencing strategy, pair and 150, and data recommendation. And the sample requirement is on the bottom. If you want to check the whole community, the microbiome community, you can choose our meta-transcriptome service. So for this one, we will sequence the complete set of your transcripts from these environmental samples, such as microbiome samples that we extract from human tissues or soil or even water. So for this one, we will provide the ribosome removal library, but you can choose if you really want the directional or non-directional library. And below will be our sequencing strategy. So still we use pair and 150 and data recommendation, and bottom will be our sample requirements. So that will be everything I will cover for the transcriptome service. And the next part will be the epigenome service. This map is kind of showing all the potential epigenome services we can offer from Novogene. So even though those RNAs could be directly quantified to check their profiles. However, those epigenomic events could be involved in different regulatory process, such as the methylation level or the interaction between DNAs or proteins, interaction between RNA or proteins, could all involved in those gene expression regulatory processes. So our service will cover those 3 different parts as well. First will be the whole genome bisulfite sequencing. WGBS is the gold standard for the methylation sequencing, and it could be used to identify the methylation at a single base resolution. And if you need the analysis from Novogene, we do need the reference genome available, and we can only perform the analysis on diploid eukaryotic species. For the library, we'll construct the bisulfite sequencing library. This is quite special for the methylation library, and we will discuss the details in the next slide. And sequencing strategy is still pair and 150, and we will recommend at least 30x for this kind of service. So using 30x is kind of describing the sequencing depth, because now we are talking about the DNA sequencing instead of the RNA sequencing. And bottom shows the samples requirements. As you can see, our sample type is the genomic DNA because we want to check the methylation of our DNA. If you check this amount requirements, it's kind of quite high because for most of those studies are coming from the cancer or disease field. And for those kind of research, they will only collect very limited starting materials from cell-free DNAs, FFPE samples, or low quantity of genomic DNAs. By using our regular or traditional WGBS method, as shown on the left, after the DNA fragmentation and the end repair, A-tailing, and adapter ligation, we will move down to the bisulfite conversion. So from this step, we will distinguish the methylated C versus the unmethylated C. But the bisulfite conversion will introduce a lot of DNA damage. So through that, from this step, we will lose a lot of input DNAs with adapter attached. So if you lose a lot of input, then we do need a very high input, initial input, to make sure you got enough data. So that's why we do need very high input for the traditional WGBS method. To overcome this question or this problem, now we propose the low input method for the WGBS. So this will be the method PBAT, post-bisulfite adapter tagging method for the library preparation. And still we start with the DNA fragmentation, and after that, we'll perform the bisulfite conversion first. So through this step, you will find out which is methylated C and which is unmethylated C. And after this step, we'll move down to the adapter ligation. So even though this may introduce some DNA damage to our input DNAs, however, we can ligate the adapters as much as possible to our converted input DNAs. So this will help us save a lot from our input DNA and increase the efficiency for the library preparation. And then we move down to the PCR amplification before our library sequencing. So with this method, it could reduce our input significantly, which I will show in the next slide. But before I move forward, I want to emphasize, if any of your samples has gone through this PBAT method, those samples could not be analyzed together with the samples using the traditional or regular WGBS method, because those two methods are quite different pipelines. And this, if you analyze those samples together, this may introduce some bias in your results. So now let's see how we reduce the input requirements. So below is the regular sample requirements, and we do need at least 2.5 microgram. But if you check the PBAT, sample requirements. So for some of those samples, we only need 20 nanogram as the input. So this will help you save a lot of your samples, very precious samples, and also perform the methylation study for your research. And the last services I want to introduce will be the IP services, including the ChIP-seq and RIP-seq. So those IP services will look into the interaction between macromolecules, such as the DNA or RNA interaction with the proteins. And for ChIP-seq, it's used to study the interaction between DNA and protein. This could be used to find out the transcription factor targeting region or some histone modification in your samples. While by using the RIP-seq, we will study the RNA, the interaction between RNA and protein, and this may deliver some potential RNA stability and structure information or the microRNA targeting region information in your research. So here I listed the sample requirements for different services. So even though we may ask a very high input of our either DNA or RNA, however, we have a lot of successful cases by using 1 nanogram of DNA for ChIP-seq or 10 nanogram of RNA for RIP-seq. So if you cannot provide those much of DNAs or RNAs, you can always contact us to see if we can try with those, if we can try with your very limited input samples. And it's quite possible we also succeed with your projects. Well, by using the ChIP-seq and RIP-seq, we do need you to send your samples with the IP and input as pairs. So IP will be your IP'd DNA or RNA, and input will be the backgrounds or the DNA before immunoprecipitation. So those will be function as the background. And if you compare your IP versus the background, that will give us the information which region of your samples has been enriched or which region of your DNA or RNA has been enriched in your samples. And with that, I'll move down to the last part, which is the case study. And if you think about our RNA services, actually, most of our transcriptome services could be used for the quick screen of your research, such as you want to find out the gene expression profile between your samples or within different samples. Or in some of the research, they may identify some biomarkers for different diseases. Like shown in this example, they found the new fusion gene, which is different from those two original genes, and this fusion gene could be used as a biomarker for the colon tumors. And also, as shown on the right, you may find some differentially expressed genes or pathways which is specifically associated with the phenotype. So all of those could be used for your quick screen, like which genes I should start with in my project. So here I list a case by using our long non-coding service. As I mentioned, for our long non-coding RNA service, we will include the information for both messenger RNA and long non-coding RNA. And this research is kind of sequencing the long non-coding RNAs from the mouse brain. And left 3 column is kind of showing the treatment group, and the right 3 column is kind of showing the samples from the control group. It's very obvious you see different patterns of the RNA expression between groups for both long non-coding RNA and messenger RNA. So by using this kind of service, they have identified 97 upregulated long non-coding RNAs and more than 100 downregulated long non-coding RNAs. And in this table, we also listed the potential target coding RNAs or coding genes, protein coding genes for different long non-coding RNAs. So this could be delivered by using our target prediction step. Our second case is kind of showing the service of the whole transcriptome sequencing. As I mentioned, so this service will deliver the information for long non-coding RNA, circular RNA, microRNA, and messenger RNA. So by using this service, they have identified different numbers of differentially expressed transcripts. And by using the network study or our free joint analysis, they also look into the network between 3 types of RNAs. And here is kind of showing their potential interactions between microRNA or among microRNA, coding RNA, and long non-coding RNAs with their upregulation information and downregulation information. And the last case I want to share is kind of the joint analysis between our epigenomic service and transcriptome service. So in this one, they want to check the gene expression levels for apple trees. So those genes are actually very important for the flowering time of those apple trees. And they check the methylation level, which are presented in those dashed line. And they also sequence the RNAs, and which could be presented by those green lines. And if you see their abundance of methylation and also RNA expression, you will see their correlation between each other. Like showing for those genes showing on the top, once they have got very high methylation level, they kind of got relatively low RNA expression abundance. So this could be used for you to explore the regulatory mechanism between your RNA and DNA. So with that, before I close my presentation, I want to share you this exciting promotion. So now we are offering the Back to the Lab promotion for whole genome sequencing, whole exome sequencing, promate library sequencing, and RNA-seq sequencing services. So if you are interested in those services, you can always feel free to contact our local representatives. So with that, I'd like to thank all of you again for joining us. And now I can take any questions.
Thank you for your insightful talk, Wei. Brilliant as always. I can see from the Q&A section that we have a lot of interesting questions. So without further ado, I would like to pick a few at random. Let's just start with a question from Ari Smith, which I actually get a few times a week, which is, can I still use your service if my samples do not meet your requirements?
It's quite a general and broad question. So we do have a lot of low input methods for different services. Like for our RNAs, if you don't have enough RNA input, we do have the smarter amplification method to enrich the template during the library preparation. So that is quite possible. And for such as I mentioned in the ChIP-seq and RIP-seq, we do have a lot of successful experiences by using quite limited input for different services. So maybe we can ask our local representative to follow up with you to see your detailed information of your project, and we can discuss how possible we can run with your samples.
Great, thank you. Next question by Nikhil Gupta. She asks, if you need to account for repeat elements with poly-A tail as part of mRNA sequencing, you need to have directional library. By large, directional library is more informational than non-stranded. What are your thoughts?
So as far as I can see, for the RNAs with very large repeat elements, so it's kind of our iso-seq will be a better choice versus the NGS platforms. But for the iso-seq service, we really don't have the directional library method currently, but the directional kit is available in the market. So if you are interested in this part, you may prepare the pre-made library, and then we can discuss how we can move forward with that.
Fantastic. Next question is, what information can I get from the joint analysis?
For the joint analysis, I'm thinking that you're asking about the whole transcriptome sequencing. For that, we will deliver the potential network, like how the differentially expressed microRNAs is corresponding to the differentially expressed messenger RNAs, and how those two components is corresponding to the differentially expressed long non-coding RNAs or circular RNAs. So it's kind of showing the potential correlation among three types of RNAs. And with that, we will deliver you the abundance table as an Excel file. And we can also deliver the potential input file for you can input them or load them to the software where you can see their potential protein-protein interactions or their potential interactions as I have shown in the case study.
Fantastic. Next question is, what about the analysis of metatranscriptome, which we did not discuss?
Yes, because we have limited time, so I cannot include all the detailed information for all services. Well, for the metatranscriptome study, we actually focus on the whole set of the transcripts you extracted from your samples. And by using that, we will absolutely construct the metatranscriptome, and we will perform the taxonomic annotation, like to annotate which species is present in your whole community with their abundance. And we will also quantify those genes we have identified in the metatranscriptome and perform the differential expression for those genes as well. So with those information, you will generally answer two questions. First, which species is active in my home community? And with those species components, what kind of genes or pathways or biological process are so active in my whole community?
Thank you, Wei. Another question is by Ari Smith, which is, do you provide single cell RNA sequencing? How many cells do you need as input?
So this is kind of a yes and no question, because for single cell RNA sequencing, for single cell RNA sequencing projects, the cell activity is quite important. So usually we cannot provide the library preparation for single cell RNA-seq projects. And for the input sample recommendation, it really depends on your sample types. Like for the cells with very high activity, we may need a little bit less, but if your cells are with very low activity, you do need a lot of input cells to construct a very good library. But for the detailed information, I cannot tell you very clearly right now, because it's really based on different cases. So maybe our local representative will follow you up after the webinar so that we can discuss the details.
Great. Next question is by Amit Roy regarding our post-data analysis support. The question is, do you provide support to customers if they have problem understanding the data analysis pipeline?
Yes, absolutely. So I will recommend different solutions. So first, we will always have a very professional sales team and technical support team in our company. So if you have any questions, even though this project has been completed already, you can always contact your TS, your technical support, for any questions regarding your projects. And also, we have all of our webinars available on our website. And one of our previous webinar is kind of introducing the detailed pipeline and analysis contents for RNA-seq. So if you are working on RNA-seq, you can always go back to our website to check the details. And if you do need other support from either TS or from other bioinformatic engineering, so we can always arrange a telemeeting or or video meeting to discuss the detailed information or detailed steps for you regarding your analysis pipeline.
Thank you very much for those excellent answers, Wei. I believe our time is coming to an end for this webinar. If we haven't got to your questions, please rest assured your local representative will get in touch with you shortly via email. Thank you for all your time today. We hope you enjoyed this webinar and found it useful. I just wanted to let you know that we will email you a link for the recording of this webinar as well as the slides. You will also receive a link for a short survey, which we would greatly appreciate if you could please complete. This would help us better tailor our future webinars to make them even more relevant to your research goals. In the meanwhile, please stay in touch. You can follow us on Twitter or LinkedIn or get in touch with us using our website, novogene.com. Thank you for your time and goodbye.